Yeah, thank you so much, Tasha and Alex, for inviting me to talk to you today. And as you can see from the title, I'm just going to cover the development of forensic DNA profiling. It's about 40 years now, a bit more than 40 years since the first demonstration. But really the key year really was 1985, and I actually joined the Forensic Science Service in the UK in 1982, and I spent those 3 years working on protein markers, because DNA didn't exist then. But what we did have were protein markers like blood groups, enzymes called phosphoglucomutase, and but there were quite serious problems with these markers. First of all, they were subject to degradation. They didn't really last very long. If you had a blood stain, even if it was dry, you wouldn't really get much of a result after a few days. The most serious problem, of course, is low discriminating power. If you think of a blood group like ABI, then maybe 10% of the population have got blood group A, for example. So again, not very discriminating. Perhaps the most serious problem was that it wasn't possible to analyse semen vaginal swabs, or it was very difficult, because a semen vaginal swab is predominantly female DNA, and with protein markers it's very, very difficult to actually see the male component. So rape cases were quite a problem. And then in 1985, Alec Jeffreys published this paper on hypervariable mini satellites, and it led to considerable interest. There was a lot of media interest, and he noted that, well, maybe this would be useful for forensic purposes, but the problem was at that time that they only had proof for pristine samples, fresh blood, not blood stains. Everything was completely fresh. So he showed that it worked really well for paternity cases, for example, but again, that was with just fresh material. So the big question was whether it would work on the sort of samples that we're interested, which are degraded forensic samples. So I started collaborative work with Alec Jeffreys in around about July 1985, and early work showed promise. And at that time I was working the Home Office. It's a government institution in the UK, at a place called Aldermaston, which is in the south of the UK, not too far from London. And we had good support at that time from the Home Office, so we had the funding to actually build a laboratory which was able to actually run these techniques. And our first publication was actually very quick. So between July and by December, we had a good proof in principle that the method was actually going to work with the sort of samples that we were interested in. So it was a very fast project, which we published in this paper in Nature, the forensic application of DNA fingerprints. So this is the very first illustration, and this is really the first autoradiograph which was produced. This is an X-ray film. We used radioactivity in those days. It's a very, very messy technique. I won't go into the details, but it would take about 2 weeks to produce one of these things. We didn't have the companies that we have today, Applied Biosystems. All these companies didn't exist, so we had to literally make all our own equipment out of bits of perspex, and we had to make all the reagents ourselves as well. And because these probes are grown up in bacteria, we literally have to have a microbiology lab as well in order to actually make the probes or the reagents which we were able to visualize. So this is what a so-called DNA fingerprint looks like. That was an expression coined by Alec Jeffreys. We don't really like the term fingerprint because it implies it's unique, and we like to avoid that expression, so we always call it DNA profiling. But anyway, these are early DNA fingerprints, and here you can see the proof in principle. It's a bit difficult to see this, but basically we have 5-year-old semen stains, 4-week-old one that compared with a fresh semen stain from the same individual, and then we've got whole blood and blood stains going up to about 2 years, and fresh hair roots here. So most of the samples which we were interested in are represented. Over here we have fresh blood, fresh semen, semen stains along here as well, all several weeks or years old. So that was the first proof in principle. But there were really 3 things which made the DNA profiling revolution possible. First was the multi-locus probe invention, which I showed you in the previous slide, which dramatically increased the discriminating power. So rather than reporting a likelihood ratio in the order of 100 or 200 if you were lucky, we were now well into the millions. So it was a highly discriminating technique. The probe labelling method was highly sensitive, although not sensitive by today's terms, because you would still need a blood stain of the order of 2 or 3 centimetres in diameter to be able to get a result. But compared with other techniques available at the time, that was considered to be extremely sensitive, because of nothing by today's standards, but in those days it was really good. The third innovation was the development of the extraction method, and we did that in our lab in Aldermaston. And this innovation was developed to separate sperm from vaginal cells. And those of you working in the area will be aware that sperm is sulfur-rich protein coats, and that protects the sperm from digestion with the addition of detergent. It makes the sperm very resistant. Whereas the vaginal cells are immediately, the DNA in the vaginal cells immediately goes into solution. So by using a 2-stage analysis, we just add simple detergent to the extract. We destroy the vaginal cells. Then we're able to literally spin the sperm cells into a pellet, give it a wash, and then come in with a reagent, dithiothiazole, which disrupts the sulfide bonds in the sperm, and then just go through the normal extraction technique. Without this method, it simply wouldn't work. And the reason it wouldn't work is because there are a lot more vaginal cells in swabs taken from rape victims than there are sperm cells. So we needed a method to do this. So this was a really important development, I think, to help to solve cases of rape. And it's that this method used here, it's the only method that has remained more or less the same over the last 40 years. So it's lasted a very long time, and I think it will be used in the future as well. So historically, the next important milestone was the first case, which was reported by Alec Jeffreys in 1986, and this involved the murders of 2 young girls, Linda Mann and Dawn Ashworth, by a serial killer, and these are known colloquially as the Narborough murders. So Alec Jeffreys was approached to help with this crime. The police had a suspect called Buckland, and they were very convinced that he was responsible for the crimes, but he denied one of the murders. He had admitted to one of the murders, but he had denied that he had anything to do with the second murder. And so much of the surprise of the police, when Jeffries carried out the analysis, he found that they didn't actually match this individual. Okay. The police were very reluctant to accept these results, first of all, and that's why they contacted me in the Home Office to carry out a replicate experiment. They were somewhat suspicious about the results. They didn't know whether to believe it or not. And it was fortunate, I think, that our lab had been working in this area. And we have done a lot of work to show that the technique was valid. Obviously, that's a very important part of forensic science, is to show that what you're doing is valid and not giving you results that are meaningless, and it's really important to be able to demonstrate this in anything that we do. So we carried out duplicate analysis, and essentially we confirmed the Jeffries result. But then obviously there's a problem. There's a serial murderer on the loose, and without doubt, he would have continued to commit these crimes. Local communities were obviously very upset about this, and that provided the motivation to carry out the world's first mass screen. Now, this murder was carried out in a fairly on a footpath, and the police were fairly certain that the individual must have local knowledge. And so they believed that he must have come from, the perpetrator was likely to have come from one of 3 villages, of which there was about 5,000 men. So these 5,000 men were invited to give blood. It wasn't compulsory. There was no compulsion. It was all voluntarily done. But obviously, if someone refused, then they would become a person of interest. So there was quite a big incentive, I think, for individuals to cooperate. At that time, the DNA fingerprinting method was very labour-intensive, and as I said, it took about 2 weeks to get a result, and you couldn't get many samples through. So we worked on this for around about 8 months. Fortunately, we had blood and enzyme profiling, which we were able to eliminate about 90%. And then we had about 10% of those 5,000 went for DNA fingerprinting. That, however, after about 8 months' work, which was required to analyse these samples, actually none of them matched. None of them matched at all. So effectively we had exonerated all of these individuals. And it's worth pointing out, I think, that all of this work was carried out at the Forensic Science Service labs in Aldermaston. But eventually, a year later, after we had apparently reached a bit of a dead end, and they were wondering whether to actually extend the search further, and this was all under discussion at the time. But then, coincidentally, in a pub, a work colleague, overhears a colleague, Ian Kelly, bragging that he gave a sample on behalf of his friend Colin Pitchfork. This individual reported this to the police, so they immediately went to investigate this guy, Colin Pitchfork, and they found that he had actually persuaded this work colleague to give blood instead of his place. So obviously they forged a passport or some ID which fooled the police. So instead of getting his DNA, they actually got this work colleague's DNA instead. Colin Pitchfork, his blood was taken, and he immediately confessed to the crime. and he was convicted. So that was the first DNA conviction anywhere in the world. I think it's worth pointing out that because this individual pleaded guilty, we didn't have to go through all of the process of was the method validated or fit for purpose. Okay, and I think that's an important point as well, because had he pleaded not guilty, then I'm not so sure that we would have had all the validation in place to support the conviction. But fortunately we never got into that position. But it was an important case because it was the demonstration of the proof of innocence of an individual, and it's pretty certain that he would have been wrongfully convicted because he had confessed to one of the murders. It was important because it was the first mass screen, and so the idea of the national DNA database naturally flows from these events. The idea that you can actually screen a large number of people, not to convict them, but to show that they did not deposit the crime stain, or they did not contribute to the crime stain. That's the important feature. So it demonstrates the power to eliminate large numbers of individuals from investigations. So that's where the seed of the National DNA Database came from. Multi-locus probes were not used for too long in our laboratories. As I said, they're very labour intensive. Reproducibility is an issue. These are, quite sensitive to small experimental differences. They're not suited for databasing because they're too complex. It's very difficult to make a database out of something like this. You can see all of these bands of various intensities, etc. And we found that many labs in Europe and especially in North America, they just simply bypassed multi-locus probes in favour of single-locus probes. So the second stage of the technology was single-locus probes, which I'll describe. But to explain where these ideas came from, we actually have to go. We can go back to 1980, 5 years before the Alec Jeffreys method, and we have this paper from Wyman and White from Massachusetts Medical School, and they showed these hypervariable regions. So unlike the multi-locus probes, these are just single-locus probes. We're just analysing a single locus. So each locus obviously is either homozygote with one band or heterozygote with two bands, and you can see that they're in all of these different positions here. So maybe if these individuals had pushed the technique or realised the implications of what they had done, then maybe the DNA fingerprinting revolution could have actually started 5 years earlier than it did. Although I have to say that the methods which were being used were not very sensitive compared to the method that Alec used. But I like to mention these things because you can see it's not just one person who comes along and invents everything. We look at the whole, what other people have done, and we build upon that. We build from many other workers, and I'll have more demonstrations of that. So these are the single locus probes. These look much nicer. We have a development here. We've got a sizing ladder, so we know how big these things are. And of course, now you're getting to the stage where you can maybe start thinking about databasing, because so long as you know the sizes of these alleles, then you can just plug those numbers into your database. And of course, you have to have several of these. Just one locus is not sufficient. So we would have about 4, and then that would give us quite a high discriminating power in the millions. So the advantage of this system was that it was more reproducible compared to the multi-locus probes, and in fact this was used in casework for about 5 years, this method. Still using radioactivity, which is a big problem, and we're still having to build these probes ourselves because we can't buy them from a manufacturer. We have to actually make them ourselves, and that's why we still need to keep a microbiology lab up and running so that we can build these probes ourselves. Biggest disadvantage with this system is that these alleles are measured in kilobases, thousands of bases. So again, there's a limitation with degraded samples. So the systems of today, we will be thinking of maybe 100 bases, for example, which gives you a much better chance of analysing degraded material. So the issue with this is it's not well suited to highly degraded samples. Okay, so yeah, by this time there was a lot of research going on in North America, and my colleagues Bruce Bedoli, FBI, and Ron Forney, RCMP in Canada, were at the forefront in their respective jurisdictions, but their main effort was on SLP rather than ML. They never used the MLP method. And the first casework in the US was undertaken by a company called Life Codes, and they started reporting cases in 1987 using this method. But then it quickly led to, this is where the defence community starts to get interested, of course, because quite rightly they need to make sure that that the techniques that we are working on are valid and are meaningful, etc. And as I already mentioned, the importance of carrying out validation. So this case is a famous case, Castro case in 1987. When it went to appeal, it precipitated what is known as the DNA wars, which between roughly 1989 and 1994, and we have famous geneticists in the US getting into the debate. Eric Lander, very high-profile figure who disputed the results, mainly on the population genetics. He thought that there was much more substructuring. We should be taking account of substructuring of populations. But I think he was probably right. The need for standards has been overlooked, and of course in our field we must always make sure that we have the correct standard. So this went on for about 4 years, dispute, and eventually we have another commentary in Nature appearing, DNA fingerprinting dispute laid to rest, and Eric Lander and Bruce Bedoli joined forces to agree that to map out a way forward. This led to 2 reports by the National Academy of Sciences, and actually the first report itself was highly controversial, and it was very heavily criticised by population geneticists and statisticians. So they had to write a second report. But the thing is that the DNA profiling got considerable attention from outside the forensic community. It was very, very high profile. And although it's I guess it's an uncomfortable time to go through this, it's actually really important that forensic techniques are challenged in this way, and I think this is why DNA profiling is as robust as it is, because of this considerable challenge that we had. It really forces us to go through the validation and to publish our results. Extremely important. It's extremely important that everything is transparent, of course, and that was greatly motivated, I think, by the events between 89 and 94. Okay, so what happened after 1989? Well, again, I'll sort of continue the theme of what other people are doing in the field. We have this invention of polymerase chain reaction, and that was again 1985. So a lot of things were going on in 1985. So you may ask, well, okay, so why didn't we go straight to polymerase chain reaction? Because obviously that's what we're using today. So why didn't we do it in 1985? Again, PCR started a new era in molecular biology, and Carrie Mullis, received a Nobel Prize for this invention because it was not just forensics, of course, but as a whole of medical genetics benefited from this. But the reason that we didn't get into this straight away is because it was quite difficult to use. So the polymerase used to extend the DNA was not thermostable. Okay, so what that meant is that you go through your cycling PCR cycle, and as soon as you denature the DNA at 95 degrees, it just destroys the enzyme. So what you have to do then is you have to literally open the tube up, put more enzyme in, and then go through the annealing, extension, and degradation cycle. And not only that, but because you need 3 different temperatures, water baths around about 30 degrees, 50 degrees, and 95 degrees. We didn't have any machines for that, so what you had to do is you literally had 3 water baths and you just moved your tubes from one water bath to another, just going like that all the time, and then when you got to that water bath you'd open up the tube, put more enzyme in, and so it's completely impractical really for forensic purposes. But then again, if we go back to this paper was written in 1976 now, and we have this paper of deoxyribonucleic acid polymerase from extreme thermophile Thermus aquaticus. So these are bacteria which live in Icelandic springs, coincidentally around about 95 degrees. So these enzymes are thermostable. So if you now take this idea and add it to that idea, now you have a method which you can use, because you no longer have to open the tube to put in new enzyme, because these thermostable Taq polymerases actually are really important for driving the method. So around about the 1990s, the technology was maturing, and Taq polymerase was used to facilitate PCR. And then we could think about the first examples with short tandem repeats, PCR in forensic applications. So I'm going to move now to short tandem repeats, are obviously facilitated by PCR, and the advantage of short tandem repeats is that they are of the region of 100 bases or so instead of kilobases. So now we can start thinking of using short tandem repeats to help with degraded samples. So here's an early example. Erica Hagelberg, Alec Jeffreys analysed the skeletal remains of a murder victim by DNA analysis. So they analysed bone, about 8 years and 13 years old, respectively. Yeah, so the second one was the remains of Joseph Mengele, a war criminal, Second World War. whose remains were recovered from Brazil, where he had escaped to. So these bones were analysed, but they used a method known as dimeric STRs, and these are short fragments, so they're highly suited to highly degraded samples. There was no multiplexing at the time, so again, you can only do one locus at a time, still using radioactivity, and we're still not at the stage where we can use, where we can start thinking of using this for databasing. So the main problem with the technique they were using were these dimeric STRs, so they're simple 2 base repeats. And the problem with it is they produce these huge number of stutters. So if you can imagine, those of you working with STRs will be aware that maybe you get 1 or 2 stutters, but you can see this with dimeric STRs, you have this huge ladder effect. So there's just one allele here, but you can see that there's multiple sub-alleles or stutters which are appearing, which makes the interpretation quite difficult. And you can certainly imagine that you would have a really hard time trying to interpret mixtures, for example. It would just be a complete mess. So these dimeric STRs, they were the first ones which were used, but again, they're not really much use for us in routine forensic work. So I want to talk more now about how the modern DNA profiling develops, and I want to talk about the birth of national DNA databases. And that work was really carried out between 1989 and 1995. Now, to create a national DNA database, or to create the tools which are used for a national DNA database, you need essentially four building blocks. Remembering that the method, if it's going to be used to create a national DNA database, it's got to be simple, easy. Really, you can't really do it with radioactivity. You have to avoid radioactivity. It must be faster, must be quite fast as well, and it's also got to be cheap. So these are the sort of criteria that we were looking for. So to achieve all of that, you need four building blocks. So the PCR with the attack polymerase and a platform on which to actually visualize the DNA is needed. Fluorescence is used to replace radioactivity. And we need tetrameric STRs, so there's 4 base pair repeats, because it was shown that these STRs have limited artifacts and stutters, so they're much, much better placed. And you recall that the examples I showed you previously were 1 locus at a time. So that's a lot of work. But if you are able to combine loci, then that reduces the amount of work required considerably, and it simplifies things. So multiplexes are important. Of course, multiplexes are achieved by having different fluorescent dyes for your loci. So the fluorescence was invented by a company called Applied Biosystems in 1989. So around about the end of the 1980s, you could see that the commercial companies are now starting to take an interest. Okay, so this is where the companies start to come in. As I mentioned, that we had to build our own equipment, etc. But now we have companies like Applied Biosystems coming in, and they are able to produce the equipment, and they're able to produce these DNA signals without using radioactivity. Okay. Still, these examples are not multiplexed, so we haven't quite got to multiplexing yet. So where did the tetrameric idea and the tetrameric STR idea and the multiplexing idea came from? Well, really this was came from Tom Caskey's lab, Howard Hughes Medical Institute, and they had the first examples of multiplexing and combined with size ladders here in this proof of principle here, and that was published in 1991. So this is an early example of using the applied biosystems technology with dyes and combining loci together. Okay, so around about that time, I was approached by a Russian scientist called Pavel Ivanov, and at that time we were friendly with the Russians because they'd recently they'd opened up quite considerably, and the Romanovs, the last Russian royal family, their remains had been discovered some years previously, and it wasn't until around about 1993 that the discovery was able to be announced to the world, but the Russians lacked the capability to carry out the necessary DNA analysis. They'd done an original anthropological analysis which suggested that this was the Romanov family, but we were approached in 1993 to carry out the DNA analysis. So it was actually it was quite contentious, and I would argue that it was actually quite an important milestone for us, because we were able to use the case to demonstrate proof in principle. At that time, about 1993-94, there was no real agreement that we should use STRs throughout the world. In fact, the US were going in a different direction. They wanted to use a different system where actually the loci, the sizes were much larger than STRs. It was a sort of hybrid between the SLP and STR system that they wanted to use. So there wasn't any real agreement that we should use STRs. Around about that time, we'd also been working with sort of low template analysis as well as mitochondrial DNA. So we had all of these techniques in our lab, and we still hadn't got around to multiplexing, though. But we were using tetrameric STRs. So there were a number of things coming together here. The short tandem repeats, tetrameric loci, and low template analysis. And coincidentally, we found that in Nazar that he had this phenomenon known as heteroplasmy, which I'll come back to. But the issue for us was that heteroplasmy had never been observed in the non-coding region. It had been observed in coding regions, but it never had been observed in non-coding region. And this caused a problem, because we had to be sure that it was a real genetic phenomenon rather than a contamination event. The case was important because we had to develop principles and guidelines that form the basis of today's laboratory practice. Controls, duplication. Duplication was carried out with another laboratory. It's Erica Hagelberg in Cambridge. Sterile rooms, etc. Because contamination is a real possibility when you're analysing these kind of samples. But what the Romanov case did do is it established the use of tetrameric STRs as the preferred workhorse of DNA profiling. So those of you who are familiar with these techniques, you'll recognise some of these loci: VWA, THO1, F13, FES, ACTBP2, that's SE33. And the majority of these loci are actually still used in national databases worldwide, but this was really the first application of these loci. So the first issue is you have a series of bodies. You've got nine bodies. You have the various servants. We've got 3 servants and the Dr. Botkin in the mass grave, and then you have 3 children, the Tsar and the Tsarina, or the putative children and putative Tsar and the Tsarina. And then it's really just a matter of just doing a paternity analysis to see if you have a family group present. So, for example, you can see that the family for VWA, they all share 15, 16 alleles, and we could go through these. I won't go through them all, but you could look at the Tsar here as a 7-10, a Tsarina as 8-8. So you can see that the children have inherited one allele from the mother and the other from the father, and this was entirely consistent with the proposition of parentage. And then, but of course, you could show you've got a family group, but that doesn't actually prove what family group it is, and that was the next question. So to work through this, we use mitochondrial DNA, which is inherited down the female line. So here is a genealogical chart. So we go right back to Princess Alice, who was the mother of the Tsarina here. She had a sister, Princess Victoria of Hesse, Alice of Battenberg, and then Prince Philip. And we were able to get a blood sample from Prince Philip, Duke of Edinburgh, and we showed that there was all of these individuals here had the same mitochondrial type, except we didn't have Alexei. That'll come back to Alexei, but he wasn't in the mass grave that was found. But to all the children and Tsarina, Prince Philip, they were all shown to have the same mitochondrial DNA. But then we turned to the Tsar. We did a similar analysis with the Tsar, and they'll come back to this problem here. There's the heteroplasmy. You can see here we have 2 signals at this position. We've got a C and a T, a low level. Okay. And when we looked at the relatives, they will have a T in this position, a T. These 2 relatives have a T there. So there is a T there, but there's also a C there. So the question is whether this is an exclusion or a contamination or something like that. Because, as I said, the heteroplasmy phenomenon had not been reported before in the non-coding region. So I won't go into the work that we did, but we had to do a lot of work, which include cloning, which is very labour intensive at that time. So the work took well over a year to complete, and eventually we published the results in Nature Genetics. So we solved the heteroplasmy problem by carrying out this cloning, and subsequently it was found that other relatives also had heteroplasmy as well. Nevertheless, there was a lot of controversy about this case. So Alec Knight from Stanford University published this paper, for example, and he argued that the fragments that we sequenced, 1, 2, 2, 3 base pairs, were too long to have come from damaged bones. So we had a lot of criticism like this, and there were various exchanges of letter in the scientific literature. But this was motivated because they had a finger from another relative, Duchess Elizabeth, which I think they recovered a finger from somewhere in Israel, and they analysed it and they got a completely different result. So there was lots of arguments about all of this, and I think the conclusion is that either they had the wrong finger or they had contamination. So, but still, there was quite a lot of controversy which persisted, and 15 years after the first analysis, 2 missing bodies, Alexei and a princess, were discovered in 2009, not far from the original burial site, and a complete reanalysis of all the samples were undertaken as a result. And this work was led by Mike Coble in the US. I think it's a good idea, really, that we didn't do this work, because obviously you could be criticised for actually repeating your own work. And by that time, by that time, the techniques had improved considerably, and not only that, but we had Y chromosome as well. So again, a lot of analysis was carried out. All the samples were completely reanalyzed. The heteroplasmy event that we originally found here, that was shown to be replicated on the reanalysis. And to cut a long story short, by 2011 we conclude the results have withstood the test of time throughout scientific advances. It is now time to put this controversy to rest. But even so, I'm still not completely sure that it was accepted by the Russian Orthodox Church. They seem to be quite resistant for some reason. So, but this was actually a critical point in the history of DNA analysis, because it was a very old sample, 70 years old bones. It was one of the oldest samples which had ever been analysed, and it showed that these STRs that we were using were actually very powerful in helping to analyse these materials. So it was one of the first examples of ancient DNA analysis, and we also established the quality controls which were necessary, using clean rooms, duplicate analysis, etc. Because the ancient DNA community was also getting going at the time, and they had considerable problems with contamination. And at the time I had many discussions with people from the ancient DNA community, and I think that our experience helped to put them on the right path as well. So many of the methods which we developed in this case are actually used in current casework, particularly the quality control. We use the automated fluorescence sequencer from Applied Biosystems, very, very different from what we're doing today, but these fluorescent labelled markers was a key development. There was increased sensitivity to a single cell by increasing PCR cycle number. Importantly, we have automation, reduced time and cost, and greatly increased throughput. And of course, that all comes from automation. That's how you reduce your costs and makes a national DNA database economically viable. Okay, so the first multiplexes that we reported, and at this stage we're still making our own multiplexes. The companies hadn't cottoned on that multiplexes were economically viable, and despite my many discussions with Applied Biosystems, they were very reluctant to get into this at the beginning. So all of these multiplexes had to be made in-house. And of course, about that time, we were actually supplying a lot of Europe as well with this. But we were a research lab, really, not really set up to create reagents. So it was important that the that the companies started to get on board with all of this. So this is a first example. It's just four loci. Discriminating power of about 10,000, 1 in 10,000, so much lower than that to which we are used for. And at that time, there were only 2 dyes available in the rocks standard to measure the sizes of your fragments. So it's quite that was the first multiplex which was introduced and actively used in casework, but suffered from having a fairly low discriminating power. Okay, so the next historical turning point was around about that time, because people in the US were still not convinced that this was the right way forward. So now I want to talk about the first mass disaster, and this is an incident known as the Waco disaster. And this involved a religious sect that sort of set themselves up in these buildings, and the FBI decided to storm these buildings, and that resulted in a big fire where many individuals were killed. And the interesting thing about this was that there were many children who were killed, and basically the sect leader was a father of many of these children as well. So they were all closely related. So these individuals were, so there were 76 people, 21 children, 2 pregnant women died, and you can see from the intensity of the fire that the remains were quite badly burnt as well, and so identification was quite difficult. So we were approached by the US Army lab and the FBI to help with this, and we used our quadruplex, the one I showed you in the previous slide, to help to analyse these samples, and at the same time the US used their systems as well. And we also had to kinship testing as well, because the many related individuals were present. So this was really literally the first mass disaster that we had to deal with. Reference samples came from personal belongings like toothbrush, razor, etc. Otherwise, we would have to get the reference samples from parents or siblings, etc. In order to carry out the statistical analysis. So this was a collaborative effort, and there resulted a couple of papers here in which we discussed the use of the short tandem repeat loci in order to solve this case. So the reason that this case was important is because, again, it provided an important proof in principle that we were on the right track with these tetrameric STR loci, and that this was the turning point for the US as well. Once we had carried out this collaborative work, everything went towards the development of short tandem repeats. So the Waco disaster was important. It showed that STRs could be used for these very difficult samples, and without doubt it accelerated the acceptance of STRs within the forensic community. And then not too long after that, in 1995, Home Office in the UK decided that they wanted to produce a national DNA database, and it's important to mention this was a political initiative. The government at the time thought that they needed more to demonstrate they were doing more for law and order, and they decided that the national DNA database would be a very good way forward. So this directive came around about 94, if I recollect. but I well remember the discussions at the time, because I was asked whether we could actually sort of produce a national DNA database, and I said, well, okay, but we've only got this this 4-locus system. We've got a discriminating power of around about 10,000. That's not really sufficient for a national DNA database. So of course the question is, well, how many loci do you need? And so we started off with six loci, which gave us a discriminating power in the of around about 1 in 50 million. So we had to develop a new multiplex of 6 loci, and a melogenin as well came in. My colleague Kevin Sullivan developed the melogenin marker, and that was incorporated. So it was a 7-locus system with 6 STRs, giving a discriminating power of around about 1 in 50 million. Later on, that lasted for quite some time, and around about the turn of the century, the companies started to get interested in this at long last. Again, still at the SGM stage, with the 6 loci, we were still making our own multiplexes, because the companies still hadn't recognised that there was a market. But then they quickly cottoned on when other countries started expressing an interest, and clearly there was a commercial market for them. And so the first commercial systems were SGM+, which was made by Applied Biosystems, and that will give you a probability of match of around about 1 in 1 billion. And of course, I won't go into the history of further extensions, but now we're up to more than 16 loci. That's absolutely standard now, and these new systems, 3rd generation systems, were introduced around about 2012 onwards. So you can see a timeline there. Okay, so that's a publication from the 2nd generation multiplex, which is the 7-locus multiplex STR test. That's 6 STRs. And you can see there these STRs are used today. And of course, once you start making a national DNA database, you're effectively locked into these STRs. You can't change them, otherwise your casework samples won't actually match the samples that you analysed some years earlier. 3rd generation multiplexes, it's work by John Butler especially, who developed reduced size STR amplicons as tools for analysis of degraded DNA, because always the challenge is to be able to analyse highly degraded DNA. So the smaller the STR, the more powerful it is to analyse these highly degraded samples with which are short fragment lengths. And we have a European network of forensic science institutes and European DNA profiling group. There's been a very important collaboration in Europe to help standardise these tests, because we have to make decisions on which loci to standardise on, and then we communicate that to the companies, and now they'll happily take this information and they'll generate these multiplexes, and we were in the position that we are today. So these scientific groups are really important to help standardise methods, and then we tell the companies what we want, and then they deliver it. So there's a timeline. So with multi-locus probes in 1987, single-locus 1989, and short tandem repeats came along in 94 with the quad system, 4 loci, and 95 we have 6 loci, and then the companies began to supply multimix in around about 99, and you've got a 10-locus system there, and then the new generation systems with 16 plus loci in 2012. And around about the turn of the century, we have low copy number as well, is coming to the forefront, so I want to talk about that. But of course, once you've got a national DNA database, first of all, it's used on serious crimes like rape, murder, etc. But of course, there's lots of other crimes going on, particularly burglary, in which case you would need to have more sensitive methods. But there's also a question of economics, etc. So when it was first used, it was only used for serious crime. But quickly, the police wanted to use it for other kinds of crimes where you have very low amounts of DNA, so-called touch DNA, where individuals might touch an object like a door handle or something like that, and then you want to profile the DNA from whoever touched the door handle. And obviously there are examples of many old cases which have remained unsolved, serious crimes, because the techniques are just not good enough. But as time goes along, as the techniques become more sensitive, then you can start reopening these old cases. So 1997, Roland van Oorschot publishes this paper to show that you can get DNA fingerprints from just fingerprints. And then we carried out some work to show you could get DNA fingerprinting from single cells simply by increasing the cycle number, 34 cycles. And we also discovered phenomena like allele dropouts, where alleles disappear and some alleles may appear, and these are called allele drop-in. And of course these confuse the profile because effectively your profile does not match your suspect. So now we get into the era of having to develop statistical models. And this is an early example in a paper I published in 2000. We had these phenomena, drop in and drop out, which we had characterized and defined. And to start off, we used a method called consensus reporting. And at the same time, John Buckleton formulated a statistical theory to combine drop out and drop in into a model. And then James Curran developed the first expert system which was able to analyze these samples. Low template DNA was also quite controversial. And it also led to the first familial searches. And I like to make the point of course, the Golden State Killer in the US was very well publicized as an example of using very large genealogical databases to find these individuals. But actually, we had carried out a very similar analysis some years earlier, and it's based on the principle that if you have unrelated individuals, they share few alleles, but if you've got full siblings, then they're going to share a lot of alleles. So we had this case with profile but no suspect in 2000. This lorry driver was killed by a brick from that bridge over the motorway. It went through his cab, landed on his chest and killed him. So the DNA was covered from the brick, and there was no match with the DNA database. But we carried out familial searching, and we found a number of potential relatives of the perpetrator. Okay, and this led to the discovery of a familial search which revealed the brother of the now convicted Craig Harman. So the brother was on the database, and by carrying out this familial search, we were able to find him. Okay, and I think I haven't got time to go through this. No, sorry. Okay, so I won't go through this one, but that was an interesting case as well. And just to finish off, we have, I'll just go to the summary. So the foundation stones were built between 1985 and 2000, and in the intervening period, between 2000 and 2015, I look at it as consolidation, development of large databases and new multiplexes, etc. And concurrent with that, we have all the development with Y chromosome, mitochondrial DNA, race markers, and SNPs, etc. Challenges for the next 40 years is we still have to deal with extreme sensitivity, so there's lots of questions about the activity, which led to the DNA profile, and that's really my main focus at the moment. So sorry I took too long over that. Okay.